Hello, everyone. Welcome back to science class. Today, we are starting a brand new lesson called Predicting Traits. This lesson is all about patterns of inheritance. In other words, how living things pass on their features to their children. We will learn about two tools called the Monohybrid Cross and the Dihybrid Cross. By the end of this lesson, you will be able to use a simple tool called a Punnett square to predict the traits of offspring. Let's get started. Before we dive in, let's look at what you will be able to do by the end of this lesson. First, you will be able to explain how a Punnett square is used to predict the traits of offspring. Second, you will be able to predict the genotype and phenotype ratios of offspring from dominant and recessive gene pairs. And third, you will be able to solve simple genetics problems using monohybrid and dihybrid Punnett squares. Keep these three goals in mind, because they are exactly what you will practice, and what you will be quizzed on today. Let's start with a story. In a city hospital, Daddy Willie and Mommy Celia welcome their new baby Volty, who has short, fair skin. Daddy Willie is homozygous for dark skin. Mommy Celia is heterozygous, and she has fair-skinned relatives. Mommy Celia is worried. She thinks the hospital may have mixed up their baby. Could baby Volty really be their child? Here are our guide questions. Number 1. How can we help solve the problem of Daddy Willie and Mommy Celia? Number 2. How can we predict the characteristics of an offspring? Think about these questions as we move forward. By the end of the lesson, you will have the tools to solve this mystery yourself. Let's learn two important words, genotype and phenotype. Genotype is the genetic code an organism has. Think of it as a letter combination, like RR or YY. For example, a pea plant with the genotype RR carries one round seed allele and one wrinkled seed allele. Phenotype is the trait you can actually see or observe. For example, a round seed or a wrinkled seed is a phenotype, because it is what shows up on the outside. So remember, genotype is the hidden code inside, and phenotype is what we can actually see. Next, let's learn two more words, homozygous and heterozygous. Homozygous means having two identical alleles for a trait, such as RR or RR. For example, RR is homozygous dominant, and RR is homozygous recessive. Heterozygous means having two different alleles for a trait, such as RR. This means the organism carries one dominant allele and one recessive allele. Now let's learn how we describe whole groups of offspring, using two more terms, genotypic ratio and phenotypic ratio. Genotypic ratio is the ratio of the different genotypes among the offspring. For example, 1 RR to 2 RR to 1 RR. Phenotypic ratio is the ratio of the different observable traits among the offspring. For example, 3 round seeds to 1 wrinkled seed. These two vocabulary words will come up again and again in the examples ahead, so let's make sure we remember them well. Now we get to the main tool of today's lesson, the Punnett square. A Punnett square is a simple grid that helps us predict the possible gene combinations of offspring, based on the genes of their parents. Think of it like a seeding chart. Each column shows one parent's possible alleles. Each row shows the other parent's possible alleles. And each box inside the grid shows one possible child. On the screen, you can see a sample grid for a monohybrid cross, RR crossed with RR. Across the top, we have the alleles R and R from one parent. Down the side, we have the alleles R and R from the other parent. And inside, the boxes show the possible combinations, RR, 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 and RR. Let's break this down into 5 clear steps. Step 1. Identify the alleles. Write the genotype of each parent. Remember, a capital letter stands for a dominant allele, and a lowercase letter stands for a recessive allele. Step 2. Find the possible gametes. Split each parent's genotype into the alleles it can pass on through its sex cells, called gametes. Step 3. Draw the grid. Place one parent's gametes across the top, and the other parent's gametes down the side. Step 4. Fill in the boxes. Combine one allele from the top with one allele from the side to fill each box. Each box represents one possible offspring. Step 5. Count and interpret. Count the genotypes and phenotypes in the boxes to get the genotypic and phenotypic ratios. Let's keep these 5 steps in mind, because we will use them again and again in the examples that follow. Let's try our first guided example, using flower color in pea plants. Purple flower, represented by capital P, is dominant over white flower, represented by lowercase p. In this example, a heterozygous purple plant, capital P small p, is crossed with a homozygous white plant, small p small p. Parent 1's genotype is capital P small p. Parent 2's genotype is small p small p. Now let's move to step 2 and find the gametes. Parent 1 pp can pass on either a capital P allele or a small p allele. But parent 2 pp can only pass on a small p allele, since both of its alleles are the same. Now let's fill in the Punnett square and interpret the results. Across the top, we place parent 1's gametes, capital P and small p. Down the side, we place parent 2's gametes, small p and small p. Filling in the boxes, we get capital P small p, small p small p, capital P small p, and small p small p. Let's count. For the genotypic ratio, we have 2 capital P small p to 2 small p, which simplifies to 1 to 1. For the phenotypic ratio, we have 2 purple flowers to 2 white flowers, which also simplifies to 1 to 1. This means there is a 50% chance of purple flowers, and a 50% chance of white flowers. Remember, every box represents one possible offspring, and all boxes are equally likely. So far, we have only been tracking one trait at a time. This is called a monohybrid cross. A monohybrid cross tracks one trait at a time. For example, seed shape only, round versus wrinkled. It uses a 2x2 grid, which gives us 4 boxes. But sometimes, we want to track 2 traits at the same time. This is called a dihybrid cross. A dihybrid cross tracks 2 traits at the same time, for example, seed color and seed shape together. Because we are tracking 2 traits, it uses a bigger grid, a 4x4 grid, which gives us 16 boxes. Let's try a dihybrid cross example, using seed color and seed shape in pea plants. Yellow, represented by capital Y, is dominant over green, lowercase y. Smooth, represented by capital S, is dominant over wrinkled, lowercase s. Parent 1 is YYSS. Parent 2 is YYSS. For parent 1, capital Y capital S small s, the possible gametes are capital Y capital S and capital Y small s. For parent 2, capital Y small s small s, the possible gametes are capital Y small s and small y small s. Here's a helpful hint. Because parent 1 is capital Y, which is homozygous, every offspring will receive a capital Y allele. That means every single offspring will be yellow-seeded. The only thing that will actually vary among the offspring is the seed shape, smooth versus wrinkled. Let's build the grid for this cross. Across the top, we place parent 1's gametes, capital Y capital S and capital Y small s. Down the side, we place parent 2's gametes, capital Y small s and small y small s. Filling in the boxes, we get capital Y capital S small s, capital Y small s small s, capital Y capital S small s, and capital Y small s small s. Notice that because there are 2 gamete types from each parent, 2 times 2 gives us a 2x2 grid, or 4 boxes total. For the genotypic ratio, we have 1 capital Y small s, to 1 capital Y small s, to 1 capital Y capital S small s, to 1 capital Y small s. For the phenotypic ratio, we have 2 yellow and smooth offspring to 2 yellow and wrinkled offspring, which simplifies to 1 to 1. As we predicted, all the offspring are yellow-seeded, because parent 1 always passes on a capital Y allele. But the seed shape splits evenly, half smooth, half wrinkled. Now it's your turn to practice. This time, we are looking at coat color and tail length in dogs. Parent 1 is capital B capital T small t, which means a black coat and a long tail. Parent 2 is small b small t, which means a brown coat and a short tail. Remember, capital B stands for black, which is dominant, and lowercase b stands for brown, which is recessive. Capital T stands for long tail, which is dominant, and lowercase t stands for short tail, which is recessive. With your group, work together to do 3 things. 1. List the possible gametes of each parent. 2. Build the Punnett square for this dihybrid cross. And 3. Find the genotypic and phenotypic ratios of the puppies. Take your time, work as a team, and be ready to explain your reasoning, not just your final answer. Let's check your understanding with a few quick questions. Read each question carefully and choose the best answer. Question 1. In a cross between two heterozygous pea plants, R small or crossed with R small r, what is the expected genotypic ratio? Your choices are A. 1 to 1, B. 1 to 2 to 1, C. 3 to 1, or D. 9 to 3 to 3 to 1. Question 2. What is the phenotype of an organism with the genotype A small a B small b? Your choices are A. Homozygous dominant, B. Homozygous recessive, C. Heterozygous, or D. None of the above. Take a moment to think through each question before we continue. Let's continue with 2 more questions. Question 3. In a dihybrid cross, R small R Y small y crossed with R small or Y small y, what is the expected phenotypic ratio of the offspring? Your choices are A. 9 to 3 to 3 to 1, B. 3 to 1, C. 1 to 2 to 1, or D. 1 to 1. Question 4. A pea plant with yellow seeds, Y, is crossed with one with green seeds, small y. What is the genotype of the F1 generation? Your choices are A. Y, B. Y small y, C. Small y, or D. Both YY and YY. A helpful tip: if you're not sure, try sketching a quick Punnett square on scratch paper to work it out. Let's go over the answers together. Item 1 asked, in a cross between two heterozygous pea plants, R crossed with RR, what is the expected genotypic ratio? The correct answer is letter B, 1 to 2 to 1. Item 2 asked, what is the phenotype of an organism with the genotype ABB? The correct answer is C, heterozygous. Item 3 asked, in a dihybrid cross, RYY crossed with RYY, what is the expected phenotypic ratio of the offspring? The correct answer is A, 9 to 3 to 3 to 1. Item 4 asked, a pea plant with yellow seeds, YY, is crossed with one with green seeds, YY. What is the genotype of the F1 generation? The correct answer is B, YY. If you missed any of these items, don't worry. Take a moment to review the Punnett square for that question, and think about why the other choices don't fit. Let's take a moment to reflect. Every trait you have, your height, your skin tone, your hair, your smile, came from patterns of inheritance passed down through your family. Take a moment to think, what traits do you have, and why do they make you you? There are no wrong answers here. Every trait is valid, and every trait is worth appreciating. No trait makes a person less worthy. Let's review what we learned today with 4 key takeaways. First, a genotype is the genetic code, like RR. A phenotype is the trait we can see, like purple flowers. Second, a Punnett square is a simple grid that helps us predict the genotypes and phenotypes of offspring. Third, in a monohybrid cross, we track one trait. In a dihybrid cross, we track 2 traits at once. And fourth, genotypic and phenotypic ratios tell us how likely each outcome is. They are not a guarantee for every single offspring. Great job today! Try explaining one of these takeaways in your own words before we close the lesson. Congratulations, future geneticists! You worked hard today, and you now have the tools to predict traits using Punnett squares. Before we end, here is your exit ticket. Write down 3 things you learned today, 1 thing you're sure of, and 1 thing you're still wondering about. Great work today, everyone, and I'll see you in our next lesson.